Our first speaker today is Alan Jiao, who did his undergraduate studies in immunology at the University of Toronto. He then moved to Yale to conduct his PhD with Frank Flack at Yale University, where he studied microRNAs, RNA splicing, and genome instability. Then for his postdoc, he moved to the lab of Yang Shi in cancer epigenetics at Harvard, and then subsequently moved with the lab to Oxford a few years later. Alan has done some really fantastic work on RNA processing and epigenetics, and I'm very excited for his talk today titled Systematic In Vivo Detoxification of H3K27M oncohistone Function. So thank you, Alan. Over to you.
Thanks so much, Ben. It's really great to be with the speaker today. Let me try to minimize that. That's okay. Right. So most of my postdoc work has been looking at epigenetic dysregulation in cancer in the context of these histone mutations which drive childhood brain tumors. These tumors are called diffuse midline gliomas, or DMGs. They arise in the midline regions of the brain. They're most frequently diagnosed in school-aged children, and the median survival following diagnosis is less than a year. For a long time, these gliomas were treated the same way as adult gliomas, but that started to change around 12 years ago now, when it was discovered that unlike the vast majority of adult gliomas, about 80% of the pediatric ones carry a very specific mutation in histone H3, where lysine 27 is mutated to methionine. And one of the most remarkable features of these histone mutant tumors compared to their histone wild-type counterparts is that the K27M gliomas exhibit this global loss of H3K27 trimethylation. And it's worth emphasizing that this was not an obvious phenotype. It wasn't as simple as, well, the M just can't be methylated, and so of course there's going to be a global loss of trimethylation. And the reason why it's not that simple is because humans have 15 histone H3 genes, which means we have 30 alleles, and only one of these is ever mutated in these gliomas, and yet this little bit of mutant histone is able to stop most of the wild-type histones from getting methylated. And so that was very interesting. That caught the attention of a lot of chromatin biology groups, including ours. And so just to summarize, a lot of work that's been done since then, these K27 mutant histones, they dominantly inhibit PRC2, which is the enzyme that is required to methylate lysine 27 on histone H3 to regulate gene expression. And so by inhibiting PRC2, these mutant histones dysregulate gene expression, they dysregulate subsequent cell fate decisions, and it's very likely that that's a major driver of gliomagenesis. And so one of the things I set out to do as I started my postdoc was to ask, well, how can we suppress or detoxify, for example, by rescuing the levels of histone methylation in these K27 mutant cells? And other groups have certainly approached this question as well. But most of these approaches have been sort of candidate-based. It's not quite obvious how we would set up a genome scale assay to suppress K27 phenotypes, and certainly a screen that has never been performed to saturation for these suppressors. And so that's what we set out to do. And to do that, we needed a system that was not just genetically amenable, but we needed a screenable phenotype. And we actually got lucky with C. elegans here. And so we introduced this K27 mutation into C. elegans. Just like in humans samples, these mutant animals lose global levels of K27 trimethylation, but importantly, they had a screenable phenotype. They happened to be temperature sensitive. And so these mutants would grow fine at optimal temperatures, but if you heat stress them, they became almost completely sterile. What that means is before heat stressing, we can grow up a lot of these animals, then mutagenize them, and then shift them to a higher temperature and just wait. And so after a few generations, any animal that still remained fertile probably got a mutation that could suppress K27 phenotypes. And so we did the screen quite a few times. We screened a few 100 million mutations, and we got 20 suppressors. Where do these suppressors map? So here's the 6 C. elegans chromosomes. 19 out of 20 of our suppressors map to the same histone H3 allele or gene where we initially put in the K27M mutation. If we look at where these mutations sit on the crowdium structure, of PRC2 bound to the nucleosome, where histone H3 is shown in blue and the suppressor mutations are shown in pink. What we see is that even though this was a genome-wide screen, most of our suppressors clustered within this 30-ish amino acid region at the PRC2 nucleosome interface. And this is just to show that these suppressors rescued the fertility phenotype at the non-permissive temperature. Most of them rescued histone methylation levels without really affecting the expression of the K27M mutant histone. And so what this suggested was very strong, unbiased in vivo evidence that supports previous in vitro work suggesting that the direct contact between the mutant histones and PRC2 is very important for this inhibition. So that was 19 of the 20 suppressors. What about the 20th? The 20th mapped to this relatively uncharacterized gene called UBC20. UBC20 is an E2 ubiquitin conjugating enzyme that tags proteins with the type of ubiquitin linkage that typically leads to protein degradation. So this particular suppressor mutation mapped to a linker region between the catalytic domain and a ubiquitin binding domain in UBC20. But importantly, just like all the other suppressors, even though the screen itself was not directly linked to histone methylation, the UBC20 mutant was able to restore histone methylation to about 50% that of wild type levels. We confirmed that the mutation was a loss of function because we used RNAi to deplete UBC20 and we recapitulated the rescue and methylation phenotype in these K27 mutants. What was interesting was this phenotype happened to be specific for K27 because if we do the same experiment in a histone wild type background, we don't see an additional increase in K27 trimethylation. And the same is true using a genetic mutant of UBC20. We also wanted to test functional redundancies between other E2s, but it actually turns out that UBC20 was unique because depleting any of the other non-essential E2s in C. elegans was unable to restore K27 methylation. We also wanted to ask where in the genome K27 methylation was being rescued, and so for this, we did cut and run. And so just to give a sort of genome scale view of the results, so every dot here represents the average K27 trimethylation signal in a 5 kb window along a given chromosome for each of the 6 chromosomes in C. elegans. And so this experiment gave 2 interesting observations. The first was that just like in human cells, K27 mutants in C. elegans didn't evenly lose K27 trimethylation. There are certain regions in the genome that just seem to be less susceptible to its effects. The second interesting observation was that the UBC20 mutant really just restored methylation in the regions that were lost the most. We never really got an overaccumulation of K27 methylation at ectopic sites. And so I think this further suggests that the effect of UBC20 on histone methylation is really specific to a K27M context. So how does this work? What's UBC20 ubiquitinating? We thought that the 2 most straightforward guesses would be either PRC2 itself or the K27 mutant histone itself. And because UBC20 puts on the type of ubiquitin that leads to degradation, we looked at protein levels. But actually, when we got a endogenously GFP tagged EZH2 homolog in C. elegans, and we crossed it into our mutants, and we really didn't see much of an effect on EZH2 protein levels in either the germline or the soma, regardless of UBC20 status. And we saw a similar thing with K27M. And so overall K27M levels were not affected by UBC20. We also did a cut and run for K27M to look at chromatin incorporation, but again, with or without UBC20, there was a similar amount of incorporation of this mutant histone. And so UBC20 didn't seem to be affecting K27 or PRC2 directly. And so then we just started to look at other histone modifications. And we stumbled across something we definitely were not looking for. When we tried to do a Western blot for monoubiquitination of histone H2B, and we overexposed this blot, what we saw was that there was so I should first mention that monoubiquitinated H2B was not affected by UBC20 mutants. But there was this additional band that corresponded to the diubiquitinated form H2B. And that's the band that disappeared in our UBC20 suppressor mutant. that also disappears in a catalytically inactive UBC20 mutant. And unfortunately, it also disappeared in our flag-tagged UBC20 strain, suggesting that this tag is non-functional. And in a histone wild-type background, none of these really affected K27 trimethylation levels. In contrast, if we look in a K27M mutant background, what we see is this suppressor mutant and the non-functional flag tag mutant eliminates H2B diubiquitination and rescues K27 trimethylation. We also wanted to test whether this phenotype was dependent on proteasome activity, and it didn't seem to be. And so with or without the addition of this proteasome inhibitor, we still see that loss of UBC20 abrogates H2B diubiquitination and rescues K27 trimethylation. And so what I think our data suggests is that there's this E2 ubiquitin conjugating enzyme that links H2B diubiquitination activity to K27M oncohistone function. And there's actually strong precedent for E2-dependent H2B ubiquitination in regulating histone methyltransferase activity. For example, it's been known since the early 2000s that H3K4 methylation is completely dependent on H2B monoubiquitination, and that this monoubiquitination is in turn dependent on this RAD6 E2 ubiquitin conjugating enzyme. Similarly, H2B ubiquitination also regulates K79 methylation. And so subsequent work has structural studies have shown that these histone methyltransferases do physically sense the ubiquitination state of H2B. And so given this precedent, and given the fact that 19 out of the 20 of our remaining suppressors also are predicted to disrupt the PRC2 nucleosome interaction, we think UBC20 dependent diubiquitination of H2B is also modulating that interaction. And so a possible model is as follows. The model assumes that K27M is incorporated throughout the genome in nucleosomes that are almost certainly modified in different modification states. It also assumes that not all of these states are going to be equally potent in their ability to inhibit PRC2. So what we think is happening is UBC20 is creating this diubiquitinated H2B modification state in a nucleosome that contains K27M, and that is a state that is a very strong PRC2 inhibitor, such that when we lose UBC20, we lose this additional ubiquitin mark, and we're left with a much weaker PRC2 inhibitor, which allows for strong but partial rescue of K27 trimethylation. Now, we don't know what is the enzyme or more details about this pathway in human cells, but for sure that'll be an interesting line of future experiments. So just to summarize, using C. elegans, we were able to perform a genome-wide screen for K27 suppressors, and we got 20 of them. 19 of these disrupted this directly disrupted the PRC2 K27M interaction by mutating the K27M gene. And then the 20th mapped to this gene called UBC20, which is required for H2B diubiquitination, and this also suppresses K27M phenotypes. And so with that, I would just like to thank my advisor, Yang Shi, for being very supportive for a number of different projects, everyone in the Shi Lab for just being fun and helpful people to work with, and our funding sources, especially the Defeat DIPG Foundation, as well as Boston Children's and Harvard before we moved to Oxford. So thank you so much for your time, and I would love to take questions.
Thank you, Alan. That was really fantastic. Just wonderful data, wonderful presentation. So I'll just note, people, you can write in your questions into the Q&A box, or you can raise your hand. It'll allow you to ask your question yourself. But I have a few to start off with. With the UBC20, where you've got this diubiquitin H2B, does that have any impact on H3K4 or K79 methylation?
Right. So by Western, no. But we haven't looked at specific loci. Yeah. No.
And then along the same lines, there's a question in the Q&A from Ji Young Park, who says, very interesting talk. How is the PRC1 histone marks, H2A 119 ubiquitin, is that affected by UBC20?
Right, yeah, so it's not. Monoubiquitin levels are the same. And actually, interestingly, we also saw a diubiquitinated band for H2A as well, but that didn't change
Mm,
in our UBC20 mutant.
it's really cool. And then another question I had was in your histone rescues, if I was if I caught it correctly, G34E rescued, but didn't really affect very strongly the K27 trimethylation. What do you think's
Yeah.
going on there?
Yeah, no, I don't know. But um so right, so I think one thing it suggests is at least some of the more organismal level phenotypes can be uncoupled from the histomethylation. That's one. Two, um, we saw the same thing in human cells, and so we expressed most of our C. elegans suppressors, the histone ones, in human cells, and all of them rescued methylation as well, except for this G34 mutation, which is much weaker. So yeah,
Mm,
there's certainly something interesting there, but yeah, I'm not sure what it means. Yeah.
yeah. And Maria says, beautiful story, Alan. Great to see it again. I'm wondering if you have tested the effect of some of the H3 mutations on PRC2 binding.
Right. So no, we wanted to. So I guess it's we totally can do that in the human system. For technical antibody reasons, it was uh, not quite doable in C. elegans. But yeah.
And then there's a couple questions asking how well conserved is UBC20 and is there an ortholog in humans?
Yeah. The sequence is very well conserved. The closest ortholog is UBE2K. One thing about UB2K though is it's cytoplasmic, or at least mostly cytoplasmic, in human cells. There is one report saying UB2K is able to ubiquitinate H3, um, but so it's definitely something we're looking into. We did the single most straightforward experiment, which was to knock out UB2K in these glioma cells. And the any rescue was at least not so obvious. And so there's maybe E2 redundancies we have to deal with, or just a different enzyme altogether. But yeah.
Yeah. And is that have you seen the H2B diubiquitin in other systems,
Yeah,
like in human
absolutely.
cells?
Yeah, and so this mark um, has been detected in a lot of systems, including human cells, including in tetrahymena. But I think, as far as I know, it's been detected the same way we detected it, just as an additional band on a Western.
Mm,
And nobody has came across an enzyme that would make that band disappear, and so it tends to just get lost in a supplemental figure somewhere.
mm.
But the band exists. Yeah.
And then there's a question or two questions from Isa Ozdemir. Says, this is a very interesting talk. Two questions. Where is UBC20 expressed? It's in the germline. And then second question, why do you think you have not seen the KGB1 mutation in your screen as previously published?
Right. Right. So UBC20, first question, it is, I believe it's ubiquitously expressed in C. elegans. Uh, I don't know why we didn't come across the KGB1 mutation. So right. So I should say there has been a previous uh, screen in C. elegans for K27 suppressors. Um, I don't think any intragenic mutations were reported. They did mention this KGB1 junk kinase, I believe, was able to rescue some of the K27 phenotypes. It didn't come up on our screen. They also found that KGB1 didn't restore histomethylation. We also checked by RNAi for KGB1 and didn't see a methylation rescue. So that's yeah.
Yeah, so okay, so that's very cool. I think that's the last of our questions. But if anyone else has questions, you can write them into the Q&A, and Alan, you can just respond there if needed. But thank you very much for the wonderful talk, and I'll pass it over to Sky to introduce our next speaker.
Thank you so much.